Okay, we can start for today. So today we're gonna this is the aim of the today's lecture is to let you understand the Drosophila chromosome structure. So this is X chromosome, this one, and Y chromosome is there, but usually we don't care Y chromosome because we don't put anything in Y chromosome. So just X chromosome. And yeah, with Y chromosome, it can be male. And with X chromosome, it can be female, as like human. Human also have XX for female and XY for male. And second chromosome and third chromosome. Mm, and fourth chromosome. And this, you can see the number in here, 1 to 20, 21 to 60, 61 to 100, and 100 to 102. So that is the numbering which is done by Thomas Morgan. So Thomas Morgan is the kind of the ancestor of our Drosophila research, and he got a first Nobel Prize in Drosophila genetic research. It's about the early 1910 in far before Thomas Hunt Morgan. So 1933. So that numbering is measured by Thomas Morgan. But actual their base pair is not like that. So I will let you later, but when we can use the website name is Flybase. When you watch the Flybase, they when you're searching for some specific gene, and they let you know where it is. So for example, to second chromosome left arm, and the number is using the Thomas Morgan's number is around 32 or something 30, this area, and it has its exact base pair number. So, but it's much easier to use this numbering. So this numbering is done by the Thomas Morgan. So he make a lot of mutation and he watch several phenotype. And using the genetics and recombination rule, so if those two things are far away, they their recombination can often happen between two spots. So they can measure the distance of two genes using the recombination rate. So when they made it and measure the how many times they recombinate or not, so they can tell the two spot is far away or just adjacent. If when you think about these two spots, this one and this one, very adjacent, just next to door, very hard to separate in the recombination. So, and this red dot is a centromere. So the chromosome need to attach the microtubule for the cell division, mitosis. So this spot is a central or centromere. So it attached to the spindle, a mitotic spindle is attached in this part. So X chromosome, they only have one arm. And second chromosome, they have a right arm, a left arm, and right arm. So when you just think about the human chromosome, where if we set the centromere at the center and right arm and a left arm and right arm, as like Drosophila chromosome, centromere is here, left arm, right arm, but second chromosome, left arm, and second chromosome, right arm. And the third chromosome, left arm, and third chromosome, right arm. And fourth chromosome, their centromere is here, but it's very small, so they only have one arm. So this is the whole genome structure of Drosophila. Very simple. We only care for chromosome. When we research using human, we need to care 23 pairs. But when you're using Drosophila, we only care 4 pairs. So 8 chromosomes. So when they make the gamete, egg or sperm, they just pick one of them, one of them, one of them, one of them, and egg and sperm they make, and then that process is we can call meiosis. So during the meiosis, recombination is occur. So recombination, which means that when the meiosis happen, so this chromosome and this chromosome, they are homologous chromosome. In Korean term is 상동염색체. So they usually don't meet in any time during any mitosis, but just one time meet in um, their schedule or lifetime. So that is just before the meiosis. Um, the pre-phase of the first step of the meiosis, this homologous chromosome meet together and they swapping this part to this part, so they swapping has been occurred. That is the recombination. And very striking and non-understandable thing is in Drosophila, recombination is only occur in the egg making. When sperm making, recombination is not happen at all. So that is very important for understanding the Drosophila genetics. So recombination is only occur in the female flies, not in male flies. So when you try to make some kind of the recombinant and to swap two genes or separate two genes or collect two genes in some same fly, the recombination should occur in the female flies. Okay, I will tell you later. So X chromosome, second chromosome, third chromosome, and fourth chromosome. So when you think about some gene, so gene have its location. So we call that loci. For example, fly have red eyes, red eyes. The red eye making gene is located in the X chromosome. We can call that gene name is white gene. The reason why the name of gene is white gene is because if there mutation on that gene, the eye color turn white. So there is the name is white gene, but it turns out that they make a red pigment, even though the name is white gene. So there's many name like that. So some gene, for example, some gene name is rutabaga or dunce. So in English term, that means kind of this little bit retarded people. It's kind of slang. So that indicates that gene is very important for learning and memory. So that is how we name it. And in mouse walk, so when you find some new gene, and we need to name it using the rule. So the gene name is of FX5 7 something, that kind of the non-familiar name. But in Drosophila, we don't have that kind of rule. So when you find some new gene, naming is on your duty and privilege. So you need to name it, and you can name what you want to name. So very interesting case is the gene name is Shilun, Korean term, because it make Drosophila eye very small. So that gene name is Shilun forever. So there is one aspect of Drosophila research, and another famous case is that Nanchong. Nanchong is in Korean term, cannot hear. So the gene name is Nanchong. So it's a hearing defect and temperature sensing mutant. And another case is Han. In Korean term, Chinese character term, Han means that cold. So it cannot sense cold. So there is a naming. So when you think about this chromosome structure and every loci, there's gene in there. And in the homologous chromosome, they have a same version of same gene contents, but it's not but 100% same. So for example, when this area decide the kind of the wing structure, and this area also decide the wing structure, but it can make kind of the straight wing. At the same time, it can make a kind of a banded wing. So there's a little, we can tell, homologous. So in that concept, we can tell dominant a little and recessive a little. So if two different phenotype is can make by this one loci, more dominant one is dominant a little. So if they have one copy, they they can make a phenotype that is a dominant a little. If they the specific allele that can make a phenotype, only the case, the homologous. The same recessive allele in there, that is a recessive allele, recessive phenotype. So also we can tell the term in the second slide. So this is the first slide. We can just briefly mention everything. And we have a second slide for the video, about making video. They introduced the genetic term first, so I will use that slide a little bit later. And how to select flies. Okay, what we done before, using the CO2 pad. So we anesthetize fly using this CO2 pad. We open the CO2 gas tank, and you can watch the fly using the stereo microscope and tell their shape, male or female, and their markers on their body. Okay. So, but however, the most important point what you need to learn is to understand their chromosome structure. So, for example, the Piazo gene, which is the mechanosensor, the specific channel that's sensing the mechanical stress on the body, is on the third chromosome. And second chromosome, maybe, yeah, so there is a specific area of the specific gene is located in their specific area, okay? And we gonna learn this thing, how to write down which one is on which chromosome. So for example, so this is X chromosome, second chromosome, and third chromosome, and we don't write down fourth chromosome. So, and semicolon, we segregate our different chromosomes. So first chromosome, second chromosome, and third chromosome in here. X is first chromosome, X equals first chromosome, second chromosome, and third chromosome. So chromosome number 2 and number 3, and X and Y. So we can call this first chromosome or X chromosome. So this is female fly, this is male fly. So male fly have Y on the first chromosome, X and Y. And when we just think that there's nothing, just all white type in the chromosome, we just write down plus. So just whole white type fly, male fly, plus slash Y, semicolon, plus slash plus, semicolon, plus slash plus. That's gonna be the total wild type fly. But in this fly, you can see the white eye. So white eye indicates they have a white mutant in the X chromosome. So white gene, it says that the gene have function to make a red pigment. So with the mutant of the white gene, so the eye color can be red. And however, when you watch this female fly, white gene, so it's mutated in on one homologous chromosome in the X chromosome, but they have two X chromosomes. So on another X chromosome, they don't have white mutant. So one copy is alive. So they have red eye color. It's the first chromosome. So when you just write down the first chromosome, you can tell that fly is male or female because male fly should have Y. And um another aspect is in the male fly, easy to make a homologous of the some a little because just have a one white mutant, but there's no homologous chromosome in the male fly. So only have a one white mutant gene. They have a homologous phenotype they have. However, in the female fly, they have a two X chromosome. So for making white eye in the female fly, they have a two Y, white mutant. So white/white, they can make a red eye. So this is the X chromosome. And this is the second chromosome. So VG, the VG is a marker on the wing. So maybe the name is vestigial or something. So VG is a recessive phenotype. So recessive marker. So one copy of VG don't have phenotype. But if you have a two copy of VG, they have a small wing. So in this female fly, in the second chromosome, one homologous chromosome have just Y type, and the other copy of homologous chromosome has a VG mutant in there. So their phenotype is just normal because this is recessive. So one copy don't make a phenotype. So just normal wing in there. And about the third chromosome, plus/plus, just Y type third chromosome. Nothing, no mutation at all in the third chromosome. So just white plus/plus, we just write that. And sometimes we just skip it when you write something. Or just write it or skip it. But when you want to explain, there's nothing on, no mutation on the third chromosome, you can just write this thing. So this is the kind of the adult genotype. But when they make an egg, they just pick one, two of them, pick one, two of them, pick one, two of them. So they have a separate combination because there's a probability. So two multiply two, multiply two. So they have a 8 probability in theory. So eight kind of the gamete, eight kind of the gametes can be made by the female fly. But yeah, it's a little less that because anyway, only 4. Because 4 kind of, because this is nothing. So 4 gamete is made by this male fly, female fly. And about the male fly, they have in the second chromosome, one copy of the VG mutant is a marker. So when you call its marker, that is a mutation, have a very clear obstacle phenotype. So and that help us to marking the chromosome or marking the chromosome and let us know where the chromosome is and tracking the chromosome. So I think you are not familiar with the concept of the tracking chromosome, but when we make the fly, the generation by generation, when you mark some chromosome, so chromosome is sent by the offspring and offspring. So first generation, second generation, and third generation, we can trace that chromosome using this marker. So this marker is alive. That means that the chromosome is sent from the ancestor. So there is a kind of marking. So when you put the name tag on our some stuff, so when you just mix it everything, but with the name tag, we can find out this is mine. That kind of logic. So when you're marking some chromosome using this marker, we can find it after several generation later. So that is important concept. So we use this marker to marking chromosome. That is very important concept. So and okay, this male fly have a vestigial, one copy of mutant, and one copy of second chromosome have normal, just wild type. and third chromosome just wild type. So when you think about the cross, this fly to this fly, so to think about the crossing, so we first imagine how many kinds of the gamete can be made by male or female. So third chromosome is nothing, so we do not have to consider this. So 2 multiply 2. 4 combination of the gamete can be made by female, and 4 combinations gamete made by the male. So in the female, okay, they just showing the result, but W, VG, W plus VG plus 4 combination of the egg can be made. And for male, W, VG, W plus Y, VG, Y plus 4 combination. And when they mate it, in the X chromosome, in the case of the W meet W, it can make a white-eyed female. W meet Y, it can make a white-eyed male. And plus W, red eye male, plus Y, just red eye male. 4 combination of the eye phenotype, eye and sex phenotype can be available. So these 4 kind of the offspring can be available. And how about the wing? So vestigial plus normal, vestigial, small, plus vestigial, normal, vestigial plus, it is also normal. So 1 recessive mutant and 2 heterozygote and 1 wild type can be made by this combination. And third chromosome, there's no mutant, so this possible combination can be available. Okay, mixed with this and this. So they can make this type of the red eye or white eye, vestigial mutant, and plus. So that combination can be made by this cross. So that is based on to understanding of this basic chromosome structure. And we need to write down which mutant is on the X, second and third chromosome using this way. And we can calculate expect or imagine the possibility of the offspring. Okay. That is a simple but very important genetic concept. And if this fly have a small wing, we can tell, okay, in the second chromosome, they have a vestigial containing chromosome from mother and vestigial containing chromosome from father. We can clearly know that with any doubt. So that is very important concept. So marking the chromosome, okay? And when we are showing that the female and white-eyed fly, so we clearly confirm that white mutant from the X chromosome have a white mutant from the male fly, and X chromosome from the white mutant on the female fly too. So only watching this fly, we cannot tell whether this fly have a white mutant on the X or not. But after the offspring, if they have a white color eye, the female fly, we can clearly tell, okay, the genotype of their parent. There's not just wild type, but white mutant, 1 copy is on the X chromosome of this red eye parent, red eye female parent mother. That is very important concept, okay? Without this cross, we cannot tell their genotype. So that is another concept, phenotype and genotype. So with the phenotype, we cannot tell the white mutant on their X chromosome. But using this cross scheme, we can tell this white eye is the phenotype of the heterozygote. And they have a 1 copy of the white gene on this female fly using this offspring. So that is very important concept on maybe that's Mendelian genetics, basic Mendelian genetics. But in using Drosophila, we can easily understand the rule of Mendel and all of basic, from the basic thing and quite advanced thing is all in there. So even though you want to start your research using mouse, I think understand from the Drosophila would be very helpful doing some mouse genetics, because mouse genetics is same logic is on there. So Mendel. So from the kind of the plant P, kind of very simple plant with the purple or white flower, uh, from that plant and from human and from Drosophila and from mouse, this rule is all same, the Mendelian genetic rule. So it is very effective tool to understand and to teach, to understand the principle of the genetics, principle of inheritance. And when they have this thing, it is not just hiding, but not disappear or just removed it. So when you have a kind of the mutant on your one copy of mutant in your genome, it is just hiding it, even though you have no phenotype, but your offspring can have the phenotype if your partner have the same copy so of that mutant. So there's a reason why almost all country prohibit the marry with the siblings, marry with the family, because family have a high percentage to share this kind of the mutant. And the offspring from the family member can easily make a kind of easily have a recessive phenotype. So that would be very lethal in many cases. So in every cultural background, they prohibit the marriage with families because their genome is very alike. So one copy can be two copy. So this in real life of the Drosophila, this fly not easy to survive basically because they cannot fly using this small wing. So and also white-eye is okay, but it cannot the visual function is not 100 functional because the red pigment is needed to watch the everything. So when when we usually use the white-eye fly as a kind of the genetic background because it's very convenient. But when you check some behavior data about they need to see something, vision is important for the behavior. We cannot use this fly because white eye, there's not 100 they don't have 100% functional vision. Okay, and this bar eye, I do not have to tell their vision is okay. Their vision is not okay. So that's the reason we need to understand the genetics, and it is very useful. And Drosophila is very useful tool to learn this thing. And it is aspect of the genetics and aspect of the disease model. So human is complex. And when you think about when you want to solve that kind of the complex mechanism or complex biology in human, you need to starting first from the simpler model because complex thing should be simplified to understand because we are human. We are not supercomputer. So to understand something, we need to simplify first. So Drosophila it's gonna be the good simplified disease model. So because it is surprisingly similar with human in genetic concept, genetics point of view and physiological point of view, and almost all the signaling mechanism or metabolism are just same. So yeah, its appearance is so different. So we are mammal, we are human. They're just insect, and they have a different life cycle, but the genome and gene structure and several kind of the mechanism are very similar. So we are easily using it and test it as a disease model. So when you want to find which gene gonna be the very key gene for evoking some disease, it is very good to use Drosophila as a first step. And if the Drosophila find the candidate gene, we can test in the mouse, organoid, and kind of the human in the hospital. So that is a step of the science, but somebody need to start the first step. So that would be good for Drosophila research. And that is our kind of the duty or our strong point and weak point at the same time for Drosophila research. We can do the pioneer work. But if it is not pioneer work, it is useless. So they're very controversial, very beneficial at the same time, very dangerous because we can be a great scientist or just bad scientist. Not between, not the middle, because we can do only pioneer work. And when we do some kind of follow-up study for everybody already know in other species or something, that research is useless. So for that kind of study need to done in the mouse or human. But what you need to do using Drosophila is only pioneer work. Everybody do not continue because it's too complex, very hard to handle. And in some case, in the technical issue, they cannot tell which one is important in disease. We can tell using Drosophila. So that is our duty and our privilege at the same time, our burden. Okay. Um, so yeah, it is very good tool, but it is very dangerous tool at the same time. So I have a plan to set up a mouse in the lab too, because we need to learn how to handle mouse because Drosophila has a specific tool, a specific function for the Drosophila. It is very strong for some point, but very weak some the other point. And the other point, we need to use mouse for the backup, the weakness of Drosophila. So that is a kind of the reasonable strategy. But I think it's very good point for the starting point to understand some on to see to or find kind of the unknown thing. And one of the funny stories, I published two main paper when I was a postdoc season. One is Nature, the other one is Neuron. But the funny point is, I'm the only first author of that two papers. So that is not common in recent era. So usually the one Nature paper usually have three first authors, usually, because for trying some for past the Nature's kind of the editing system, you need to present very fresh and very striking data and new finding. And for making their criteria, they need to help up to several team and then make many first authors. But in my case, there's a work, only me is the first author, both two papers. So there is a good point and dangerous point at the same time. So anyway, it is not essential story to understand the Drosophila genetics. Okay, and let's move on. I will finish around 10:30. Okay, this 10 to 12 already, but I already told that the most important part for today. So I will not demand too much in one day. And okay, so and as I mentioned before, there's a two thing in the chromosome in the fly line. First one is mutant. Second one is transgenic. So this one is all mutant flies. So mutant of the white gene, mutant of the vestigial gene. So these genetics are only depending on the mutations. But we have a transgenic. So transgenic is introduce some kind of the gene a fragment to the specific area of the chromosome. So P element which is a jumping gene. So we clone some gene using the E. coli vector and design some kind of the gene structure, gene sequence in the vector, and we move the vector to the specific vector that can go into the fly genome. I will tell you in the next session. So we in the short, we purify the plasmid and the plasmid containing our interest gene. And also we introduce the kind of transposase specific enzyme. So we inject that specific DNA fragment to the egg's germline. Egg have their germline. We inject it, and maybe some electric shock is can introduce that gene, and at the same time transposase is activated, so transposon can be activated. So transposon introduce that gene fragment to the genome, and in theory the insertion site is random. So they randomly introduce this area, but in the later technology, we can designate the site in introduction site in when you're using ATTP or ATTB landing site technique. But without that, that can be inserted in randomly. So that make another issue. When P element introduced in the genome, that P element break down some gene. That is how can I found 3 new mutant. Because first I want to use the UAS line, RNAi. So, and I got the line from the NIG from the Japan, and they make the insert the P element using that way, random insertion. And they express that the P element express some specific RNAi, so specific RNA that inhibits some specific genes function. At the same time, they landed in the random area. So I want to know where it is. And it turns out several new gene area, and it makes sleep phenotype. So not because of the RNA effect, but because of they land in the unknown area and they break down that gene. So there is a story that I found 3 new mutant when I start my graduate student. Just never expected, never, there is kind of the artifact, there is kind of the side effect. But using the side effect, I found 3 new gene, and every that candidate has been published in later, but none of them is not my paper. None of them is not my paper for my 선배 or for my 후배, because at the time, the graduate students should graduate and they need to get something, but it is hard to get some new thing in the research. So there is a story. So that is how we introduce the P element, and usually people hate it because they don't to make an unwanted mutant. But in other aspect, it can make mutant if it's fortune or unfortunate. If they insert in the just intron or just non kind of hotspot of the gene, it's totally okay. But fortunately or unfortunately, they landed in middle of specific gene, and I published 2 new papers, and one of them is very important paper, SIF amide receptor, which is important for the sexual behavior and maybe sleep behavior in Drosophila. That's the story. So around 2007. Anyway, I just because of your question, I reminiscence my old story. Yeah, there is all those things related. So you need to understand that when you watch something in this kind of the formula, mutant or P element. So this is mutant. And also we input the P element to this area we can write down P something. Okay. So there is a good things in there. You need to read it a little. I explain the concept of a little, so loss of functions, a mutant, hypomorph, or gain of function. So you can read it. And recombination. I already explained a little bit of the recombination. I just skip it. And this is explanation of the recombination. So male fly, never recombinant, no crossover made in any cases. And how about the female fly? So when you think about the one chromosome, RY mutant is on one side, is a marker, and mutant and marker at the same time. And ebony is on the other side. So random crossover can happen if these two gene, mutant gene, is located in the far away, the crossover can be made easily. And and the other case, if these two things is just adjacent, crossover will not make almost at all. So we can calculate the distance between two markers because we can, after mating, we can count the recombinant flies. So many recombinant flies indicates that they're far away. A little, very little or no recombinant fly indicate they're just adjacent. Using that method, Thomas Hunt made this kind of the map, just using that way. Because we have no tool to look into what's going on in the chromosome, but we can tell using the phenotype of the offspring. RY is a ruby eye, the eye color. Ebony is a body color. So the kind of the more darker body color they have. So they have phenotype. So usually if we assume that ruby and ebony can be on the same side of the chromosome. And when the recombinant has been occurred, they can separate. So usually ruby and ebony usually on the same flies, always. But after recombination, ebony only, not ruby. Ruby only, not ebony, can be occurred because of the recombination. Because of recombination, these two things separate, and these two things are separately transferred to the offspring. Some offspring only get ebony. The other offspring only get a ruby. But without the recombination, the ruby and ebony should be always occur at the same time. So there is a kind of the how we can find the recombination can be occurred. And this probability is around less than 10%. So if the most prominent recombinant thing is around 30%. So you can make this recombinant so to separate two things and to gather two things, one chromosome. Because separate is available at the same time, gathering is available. So you can get this, and you can remake this again using the recombination. So there is a concept of recombination. So, and this recombination is taking place just the pre-stage of the first meiosis. So before they separate of the homologous chromosome, they stick each other and they pairing each other and they switch it. So there's a reason why different species cannot make offspring, because for making gamete, the pairing should be essential. But if the pairing is not made, and their gamete is not made, the functional gamete is not made. So there is a very necessary step for making the gamete, and recombination is occur at that time. Okay, and we also have a specific enzyme to evoke the recombination. So by expressing that enzyme, we kind of activate the recombination rate. Or another point is transposon. So when we introduce some kind of the gene in the genome, P element, so there's a transposase, a specific enzyme to help the p element can introduce in the genome. And you can also express this transposase to pop in or pop out specific p element inside the genome. So how can you tell the p element insertion site? Okay, that is not in here. Okay, about the p element, I will let you the next time again because that is very essential, the jumping gene concept. But today you need to understand the concept of the mutation, mutant, and recombination, and structure of the Drosophila chromosome, and gene descriptors and locators. So gene have a name. So they we will usually use a fly-based homepage. So fly-based is the homepage. Okay, let me stop sharing. Here, this is a fly-based homepage, flybase.org. For example, page 44. DH44 is the kind of a diuretic hormone gene that secrete the DH44 neuropeptide, which is a hormone that evoke the body fluid secretion and gut movement and feeding behavior, especially carbohydrate. So when you put the gene name, symbol, species, name, diuretic hormone 44, the diuretic, which indicate that activate urination hormone 44, number 44. The CG number, CG is a Celera Genomics, the name of the company. So when you recall about the Human Genome Project, after Human Genome Project, Cellular Genomics, the company, do a lot of work in the human genomics. After that, they just tell, okay, we can sequencing all base pair, all gene or DNA of the fly, mouse, and other model system. So they do it. They have and they number it. The CG, cellular genomics, and 8348. They name it. But the DH44 is named by the another person who found the kind of urination phenotype, activate the urination phenotype of this hormone. And you can tell everything. So I'll let you know how to use this website later. But it is very useful for the understand the fly gene structure, also available fly line, how many fly lines, and especially it's also very extremely useful to study. So they summarize all the references, papers. So representative publication, all publication. So they summarize to understand the function of this gene, TH44, they can showing the kind of the representative papers and Monica's paper. So that I published a follow-up study of this paper. And all publication like this. Okay, show all. So they showing the all publication relate with the DH44. So it is too easy to find relative papers. So very simple. Download all the paper and read all the paper, especially representative publication. So when some new student join, I let how to use this website and I train them to go to the website of each papers and download the PDF file and download the kind of the citation file and give me. And I will let you do it in later, but not now, because it's only the second time. And actually what I'm doing is find my paper. Where is mine? Why is not on the front page? It's very annoying. Here. Okay. But I'm very annoyed because it's not on the first page. It's not good, not good. Anyway, okay. Just wrap up today's lecture. Okay, so finally, I will be showing this. In Drosophila morphology, there's a name on everywhere. So I showed you this yesterday, but you can watch the eye and antenna and brittle and wing and thorax and abdomen, but they have each name everywhere. For example, arista, antenna, orbitals, and um humerals. This bristle name is humeral. The bristle means the hair, specific hair, humeral, and halteria, and proboscis is the mouth of the eye, mouth of the fly, and 1 millimeter. And in the abdomen, the T1 to T to 3, it's kind of the segment, and 2S, 3S, 4S, and anaplate and genital arc, and penis in there. So anus, penis are different, and anaplate and vaginal plate is different. So, and they have a number 80, and a mayfly only have 6 to 70. So one segment is less in the mayfly. And this is a halteria. Halteria is usually insect have 4 wings, but fly only have 2 wings, and they are better fly. They fly functions better because this halteria making the balance for the body. So when the wing is moving, the halteria is also moving the opposite side, so they balance their body. They're super efficient way. They balance their body very easily. So 4-wing, a 2-wing fly better than 4-wing insect. They're better flyer. Their flying ability is very surprising. And energy efficiency is always surprising. And this wing structure, I always showed you that there's a vein structure I showed you in the kind of the 2 days ago. In the under the microscope, they have a name on each vein. So and they check whether okay, this vein is okay, and everybody discuss okay, submarginal cell is okay, and three and distal cell is okay. So they can, if somebody says that, then they understand because of reason they name it one by one. So all the name. So, and this is the several markers in Drosophila, and but I will starting from this point in the next class. Okay, so wild type, but there's a marker is a specific phenotype of the mutation. So body color, eye shape, bristle shape, bristle length, wing structure, wing size, and there is a kind of the criteria, and you can find something. There's a marker or the phenotype of mutant, and we can trace the specific chromosome using this marker. Okay, so I will finish today's lecture, but next time I will starting from this. Okay. 자, 오늘은